We have reached the final set of topics in our unit on genetics. What we're going to consider in this presentation are human genetic diseases. We're going to start off by looking at diseases caused by or associated with single genes, and then talk about some chromosome level disorders. One of the oldest tools in the toolbox that geneticists have to understand genetic diseases is the pedigree. A pedigree is essentially a family tree used to track a particular trait. Typically that would be a disease. So in a pedigree, males are squares, females are circles. If a person is affected by this trait or they have this trait, their symbol would be shaded. A horizontal bar connecting a male and a female indicates that they have had biological children. So if you drop down from that bar, you will see their offspring. And they're listed in birth order. So on the left side there, we have a male and a female that had 3 kids. Right? They had a girl, a boy, and then another boy. 2 of their kids were affected by this trait. 1 was not. The pedigree is usually created for a person, right? They might go to a doctor or a genetic counselor and they have a disease, or they're concerned about the possibility of having a disease. And they might sit down and reconstruct their family history, create a pedigree, and that will inform them about the mode of inheritance of a trait. And by mode of inheritance, I mean, is the trait autosomal? Is it found in chromosome 1 through 22? Or is it sex-linked? And is the gene involved dominant or recessive? What we see in this pedigree is a case where the trait shows up in each generation and it's affecting males and females. So that's a signature of a trait that would be autosomal and dominant. If you want some practice interpreting and even constructing a pedigree, check out Lab 11. There's a pedigree activity in that lab. We're going to start off with traits that are autosomal, so they would be caused by genes found on chromosome 1 through 22, and they exhibit simple dominance. Now, the first question you might ask is, why do we have diseases that are heritable? Now, I mean, how is it that there are diseases out there in the human population that you can inherit? You don't have any choice, right? You might just be dealt a crappy genetic hand. Why? Why are there disease-causing genes? Whose idea was that? So, diseases that are genetic are the result of a mutation in an otherwise normal important gene that creates a new allele, a disease-causing allele. So at some point in human history, a mutation occurred at a gene that created a disease-causing allele, and that can be passed on to the next generation. So in this flowchart here, we see at the top a normal version of a gene is the instructions for making a protein, and that normal protein has an important function and it results in a normal phenotype. But a mutation in that gene creates a new allele that encodes a protein that is not normal, that doesn't function normally, and that results in a disease phenotype, right? So for each gene we're going to talk about, at some point in human history, a mutation occurred and a disease-causing allele was created. Disease-causing alleles are often recessive. Often, but not always, right? So when a mutation occurs and a new allele is created, often that new allele is recessive. Here's an example of some diseases that are autosomal and they exhibit simple dominance. The first is called PKU, phenylketonuria. This is an enzyme deficiency. So people with PKU, the amino acid phenylalanine is allowed to be converted into phenylketone, which can damage the nervous system. So since it's a recessive disease, you have to be homozygous recessive to express it, and people with PKU should not be consuming foods that contain a lot of phenylalanine. This is a weird disease in that most of the problems associated with it can be avoided by diet. A second example is albinism. So this is also caused by a recessive allele, so you have to be homozygous recessive to be albinistic, and it prevents the production of the pigment melanin. Cystic fibrosis is one of the most common serious genetic diseases in people of European ancestry. It is a mutation in the gene that produces chloride ion channels in the cells of your respiratory and digestive system. So this is a lethal disease. Our last example here is one where the disease-causing allele is actually dominant. So you only need one copy of the mutated allele to have Marfan syndrome. This is a disease of connective tissues. So people with Marfan syndrome have some body-wide issues related to connective tissues. They tend to have a very tall stature, and the most serious medical problem is a weak aorta. The aorta is the main artery exiting the left side of your heart, distributing blood to the rest of your body. So I mentioned that people with PKU should not consume the amino acid phenylalanine. One source of phenylalanine is the artificial sweetener NutraSweet. It's made from phenylalanine. And there's a warning label on all products that contain NutraSweet. There it is on this can of Coke Zero. Phenylketonurics contains phenylalanine, right? So you should avoid consuming that if you have the disease PKU. There are heritable human diseases that have incomplete dominance. So remember that that's a condition where the heterozygote has an intermediate phenotype between the two alternate homozygous types. The snapdragons, right? We had red and white made pink. Our example here is a very serious and debilitating disease called sickle cell anemia. Sickle cell anemia is a mutation in a gene that codes for hemoglobin, which is the oxygen-carrying protein in our red blood cells. Anemia is a term that describes a number of conditions where you have reduced oxygen-carrying capacity. You could have anemia from a dietary deficiency, or you could have this inherited version where abnormal hemoglobin causes the blood cells to curve, right, to be shaped like a sickle. So if you are homozygous for the sickle cell mutation, you have sickle cell anemia. In this picture we see on the left normal red blood cells, on the right, the sickled red blood cells. The weird thing about this disease is it's a difference of only one amino acid in a chain of hundreds. So just changing that primary structure by one amino acid causes these cells to become misshapen and have reduced oxygen carrying capacity. Sickle cell anemia is an example of a disease where one gene has a cascading effect and causes problems body-wide. This is kind of the opposite of polygenic inheritance, where one trait is caused by many genes. Here, one gene affects many traits. That's called pleiotropy. So now the reason I'm saying this is incomplete dominance is if you are a heterozygote, if you have one copy of the normal hemoglobin gene and one copy of the mutated, you have a much less severe condition called sickle cell trait. And your blood cells only would experience abnormal shape and reduced oxygen carrying capacity under certain conditions, typically when you're in low oxygen environments or low oxygen conditions. As you might be aware, sickle cell anemia is much more common in people of African descent. In the United States, 1 in 400 African Americans have sickle cell anemia. There's a complicated, interesting history here with this gene. It is likely that the original mutation arose in Africa, right? So it makes sense that people of African origin would have that gene present in higher frequency. When we look to Africa itself, there are regions where 20% of the population has at least one copy of this gene. Now, for a disease-causing allele, that is a ridiculously high frequency. There's got to be something going on there. And there's an evolutionary explanation for that. When we look at the two maps here, on the right, that's the distribution of the parasite that causes malaria. And there's a big correlation between high frequency of the sickle cell allele and the distribution of malaria. So as it turns out, people who are heterozygous for the sickle cell mutation have a great degree of resistance to malaria. So evolution probably selected for keeping this mutation around in the heterozygous condition, even though if you wind up with two alleles, you have a very serious disease. Being heterozygote would give you a great advantage in areas where the incidence of malaria was very high. Now we're going to look at a few traits that are sex-linked. Sex-linked traits are those resulting from genes found on the sex chromosomes. And remember, in humans, the X is the big one of the two. Most of our sex-linked traits are X-linked. This is a pretty famous family portrait. This is Nicholas, the last Tsar of Russia, and his wife Maria, who was the granddaughter of Queen Victoria. And the pedigree we see here indicates that their son inherited a disease called hemophilia. Hemophilia is a disease that is recessive, and it results in the inability of your blood to clot. So what might be a minor injury can become a life-threatening condition. When we follow up this pedigree, we see his mother and his grandmother and his great-grandmother all have a half-shaded symbol. That means that they were heterozygous, right? Females have two X's. One X had the normal allele, one had the hemophilia allele. So with the recessive X-linked trait, it shows up more often in males because males inherit an X from their mom and a Y from their dad. And they only need one copy of the gene to express the trait, even if it's recessive, right? So the daughters were all normal. We don't know whether they were homozygous or heterozygous, but we know their son had hemophilia. A second example of a recessive X-linked trait is color blindness. And this is a plate from a test for color blindness. So if you have normal vision, you should see a number 6. No, just kidding, it's a 7. So color blindness, the most common form, is called red-green color blindness. So this is a mutation in a gene that produces photosensitive pigments in our eye called opsins. And the mutation here results in the inability to distinguish red and green. Here is another plate. We see a number 8 if you have normal vision. If you have red-green color blindness, those dots all are variations on the same shade, and you can't see that number 8. Color blindness is fairly common. In fact, I have a colorblind son. My wife is a carrier. She's heterozygous. Her father had color blindness. So it's common for this trait to pass from grandfather to mother as a carrier, then on to grandson. Now, if you were colorblind, there is an occupation that might be somewhat impractical for you. And my son, the colorblind artist, right? So he paints, which can be a challenge if you're colorblind. One of his jobs is to paint murals at the St. Louis Zoo. And this is the Armenian viper exhibit he painted a few years ago. Beautiful work, huh? So looking at that, you wouldn't necessarily know he has a hard time telling red from green. Those red tulips, for example. He knows they're red because the can of paint said red on it, but to him, it's about the same shade as the grass. Well, so far we've been looking at diseases caused by one gene. This is a human karyotype here, right? So we talked about autosomal genes. They're found on chromosome 1 through 22. We just looked at a couple of X-linked genes. There are diseases, though, that are associated with problems that involve entire chromosomes. So here, rather than saying a disease, we might use the word disorder, because it is a collection of problems caused by an entire chromosome or parts of chromosomes. Back in the lecture about cell division, I introduced the term aneuploidy. That's the term we use to describe a karyotype where you have an extra chromosome or a missing chromosome. 2n plus 1 or 2n minus 1. So here's a karyotype from one of the more common aneuploidies in humans. It's called trisomy 21 because what? The 21st chromosome is present in 3 copies instead of 2. This is more commonly known as Down syndrome, and this would be the karyotype from a female with Down syndrome. Now, regardless of age, gender, ethnicity, you can recognize someone that has Down syndrome. They have a suite of common physical traits because they all have 3 copies of the same chromosome number 21. Did you know there's a correlation between the incidence of Down syndrome and the age of the mother? Check out this graph, right? On the y-axis, infants with Down syndrome per 1,000 birth. The x-axis, age of mother. As you go above age 35, right, we see it go up to 10. As you go up to 45, up to 30. As you go up to 50 years old, it goes up to over 80 births per 1,000 have Down syndrome. What is going on there, right? The same correlation does not happen with the father's age. This really is a function of the differences in meiosis between males and females. This image shows sperm production. With males, sperm production doesn't start until puberty. And in the testes, there are diploid cells that are capable of going through mitosis, like sperm stem cells. And once a cell engages in meiosis, it completes it all pretty rapidly, making 4 haploid cells. In females, though, meiosis is this stop and start process. In females, meiosis actually begins while the female is still an embryo, a fetus. In utero, cells in the ovary start to go through meiosis, and they are arrested in meiosis 1 at birth. And the cells are arrested in that state until puberty, where a cell picks up where it left off all those years ago and proceeds into meiosis 2 and stops there until fertilization happens. So this big time gap and the stop and start nature of meiosis in females makes it much more likely that a non-disjunction error will happen, resulting in an extra or missing chromosome. In addition to chromosome number X, or 21 rather, the X and the Y chromosome also can be present in extra or missing counts and still have a viable fetus and a viable individual. So here we see some aneuploidies of the sex chromosomes. And one of the more common ones is Turner syndrome and Klinefelter syndrome. So Klinefelter syndrome is a male phenotype where there are 2 X's and a Y, right? So because the X doesn't carry as many vital genes as other autosomal chromosomes of the same size, you can get by with having an extra or a missing one. Turner syndrome is we call it a monosomy, so it's 2n minus 1. Turner syndrome female has only 1 X chromosome. There's no homologue. There's no partner to that chromosome. We don't see many other viable aneuploidies beyond chromosome 21 and the X and the Y. The other chromosomes just carry too many important genes to have a normal embryonic development with extra or missing chromosomes. Here's a karyotype from a rather extreme case of Klinefelter syndrome, where there are actually 4 X chromosomes, right? So again, this is still a viable condition, particularly because in human cells, only 1 X chromosome is active at any one given time. So in this individual, 3 of those X's would exist as an inactive, dense little body of chromatin called the bar body. In addition to diseases or disorders caused by the number of chromosomes, there can be disorders related to alteration in the structure of chromosomes. So if there is a mutation that occurs during meiosis where chromosomes are breaking and there's some kind of rearrangement or change in structure, and that cell winds up becoming a gamete, and that gamete's used to make a zygote, that becomes a heritable genetic condition. We have here three terms that we use to describe different kinds of chromosomal mutations. In this figure, those jaggedy arrows on the left are showing us the places where chromosomes are breaking, right? So chromosomes break because of physical stress. They break during crossing over. They can break if they're exposed to energy or radiation or chemicals. The top frame shows a deletion, where if there are two breaks and that chunk of chromosome is removed, and broken ends of chromosomes are very sticky and they want to reanneal. So you may lose a piece of a chromosome. That's a deletion. Let's say a crossover event was happening, and the crossover wasn't reciprocal. Instead of swapping equal pieces, one chromosome received a piece without giving one up. Now we got an extra chunk, right? There's a duplication. The third example, we've got two genes. There's a red one and a green one. So this is not a colorblind friendly illustration. But the red's on the left, the green's on the right. Inversion would involve a break, and the piece gets switched, flipped over, and reinserted, right? So the position of the genes has changed. And position matters, right? Genes are flanked by sequences that regulate how much protein should be produced when that gene's expressed, right? So if, let's say, next to the red gene, you had a sequence saying, oh, we don't need very much of this protein, but next to the green gene, we have a sequence that says, make a lot of this protein. So after the inversion, you could have inappropriate amounts of gene expression, too much of one protein being made, too little of another. A fourth type of rearrangement is called a translocation. And translocations are like crossover events between non-homologous chromosomes. So here we have a specific example. In humans, if there's a reciprocal translocation between chromosome 9 and 22, they're swapping pieces, right? And the piece from the telomere end of the long arm of chromosome 9 is swapped with 22. And that parks a cancer-causing gene right next to a very strong promoter. And that can result in a type of leukemia, right? So this translocation can cause a certain variety of leukemia. Here's our last slide in this presentation, and I guess the last in this genetics unit. Check out this karyotype, right? And as you look it over, there's something odd about this. Oh, look at that. Chromosome pair number 21. There's only one, right? So you might think, well, that can't be viable. Well, this person actually has a second chromosome 21. It is just attached to a different non-homologous chromosome. Chromosome 21 is so small that it can be tacked on to another chromosome, right? So there's a translocation. It looks like chromosome 15 in this image, right? So imagine the second 21 is attached to the number 15. This person's normal. They have two copies of chromosome 21. But they have a 50% probability of producing a child with Down syndrome if, during meiosis, they produce a gamete where the chromosome 15 that has the extra 21 attached to it, if that gamete also includes the normal chromosome 21, when that gamete fuses with another, the zygote will have 3 copies of chromosome 21 and they will have Down syndrome. So there's actually 2 different causes of Down syndrome. Both involve having 3 copies of chromosome 21. Most, 95% of all Down syndrome, is the hiccup that happens in meiosis called non-disjunction. 5% is familial, which means it runs in the family, right? So like a single gene might, this translocated chromosome can get inherited. So that's kind of weird. There are 2 different reasons why Down syndrome can happen.